Alright, in this video we're looking at the eukaryotic cells and the internal membranes that compartmentalize their functions. So we're talking about organelles more or less, and of course eukaryotes are the only ones that have organelles. You need to know that prokaryotes generally don't have them and eukaryotes do. Bacteria are the prokaryotes, as are archaea. There are other things that are eukaryote that are single-celled. Things like, right here, protists, fungi, and some animals that are microscopic, and plants that are eukaryotic single-celled organisms, but the majority of eukaryotic organisms are multicellular. They are made of many cells that are organized together. We are eukaryotes. The basic features of cells are, of course, they have a membrane. They have the cytosol or the cytoplasm, all right, which is kind of the goo that all the organelles kind of sit in. They have chromosomes, they have ribosomes. All cells have these things, but eukaryotes, of course, have more things than prokaryotes. Prokaryotes are very simple, all right. These are complex. They have organelles, and they have a lot more DNA, of course, to regulate all these structures. So there's no nucleus in a prokaryote. DNA is unbound region called a nucleoid, also sometimes called a genophore. You might also hear it called the bacterial chromosome, but that is a misnomer. A chromosome is single-stranded, and it's a strand, a long string. And in prokaryotes, the DNA is a circular thing, a nucleoid or a genophore. And since it's circular, it has no ends, so it's not a strand, so it can't be a chromosome. But everyone calls it the bacterial chromosome. It's a slang word, but it is technically incorrect. So here we go. This is a bacteria. So this is how we draw it in the book. This is what it looks like in reality. So you're kind of looking around there. Where is everything? It is kind of hard to figure out what everything is here. But the membrane is certainly on the outside. So the pink is probably lipids. Proteins are probably there. The DNA is probably the darker pieces, but we can't be sure of that. But the idea here is this is what we see in the book, and this is what we see in reality. This is probably with an electron microscope that we just learned about in the last video. All right, and this just is a bigger view of it. Now, the DNA in a eukaryote will be in the nucleus, and it will be surrounded by, which is made of membrane. It's a double membrane. So it's extra protective of the DNA, and the DNA needs to be protected. I will give you some more videos on that. Now the cytoplasm is the goo. It's really just water and proteins dissolved in it, and ATP and some glucose and other things just kind of floating around. And it's called the cytoplasm. And essentially the organelles sit in this jello-like material. So eukaryotic cells, of course, are much larger. It's about 10,000 to 1 at the most. The smallest bacteria, the largest eukaryotic cell, the difference is about 10,000 to 1. So the plasma membrane, this is the outer membrane of the organism, and both eukaryotes and prokaryotes will have one. And of course, its function is to keep the things out you want to keep out, and keep the things in that you want to keep in, and then to selectively let things that are useful to you, like oxygen, all right, nutrients, and waste pass through there. So you want to get rid of the waste, but you want to let in oxygen and nutrients. The one waste is CO2. You want to get rid of CO2. You breathe out. Where does all that CO2 come from? Well, inside the cell has to get rid of that CO2, so it excretes it out of the cell. It has to pass through the membrane and to get out of the cells and then into your bloodstream, and then you breathe it out eventually. And that's all about how hemoglobin works. It brings oxygen to the cells and takes the CO2, which is a waste, away. Now here's what the membrane actually looks like. So this would be these two little does it look like? That's the membrane of the cell, and this is how we draw it in the book. These are lipids here, but these are proteins. Actually, they're not single proteins. They are complexes of many proteins stuck together, and they will sit in the membrane like this, and many of them are pores or pumps that pump things in or out of the cell. So you might have something that pumps a particular chemical in or out, or a particular molecule. Now, some things have to attach, and on the outside of these protein complexes you will fit a carbohydrate. You can think of a carbohydrate as kind of like like pizza dough is a carbohydrate. If you get it wet, it's kind of sticky, or sugar. Carbohydrates, technically, they're sugars, and they get wet, they get sticky, and that's what these carbohydrates are sticking on the outside of this membrane for. They're for to attach to things, and specific shapes and specific types of carbohydrates here will allow certain things to attach and other things not attach. But the idea is they're stuck here to attract and stick to certain things, like that insulin we learned about. We learned insulin sticks to the outside of the cell, and that means that there would be a protein sitting here in the membrane on the outside of cells that is specific to attaching to insulin. And then there's a signal between, now we're not talk about it at this point, a signal between this protein and this one maybe. And so when something sticks here, like insulin, it's a signal for this one to open up and let glucose in. And we talked about that in the previous test. So here we have the outside of the cell. Now, metabolic requirements. In other words, there's so much energy to go around, and so a cell has to be, if it gets bigger, it's going to need more energy, which is in the form of ATP, which is what we do. We turn glucose into ATP, but we can eat carbohydrates and other things and turn them into glucose, and then the mitochondria is its primary role, and there's a video out there on this that is designed to convert glucose into ATP, and of course to recycle that ATP when it's used up. This is talking about surface area. Kind of be familiar with this, but you don't have to learn it in detail, but be aware of the concept. The eukaryotic cell, inside, it's going to have lots of structure. Plant and animal cells are close, but not the same. They all have an outer membrane, though. That's one key thing. This is your generic eukaryotic cell. There is no cell that looks exactly like this. All different types of cells. This is one that kind of has all the features. We're specifically going to talk about the rough and smooth ER, and that's what this green and this blue stuff. The rough have ribosomes on them, the smooth do not, and we're going to talk about those in a different video. We also you need to know what mitochondria look like. They're here. There are other organelles, which I will talk about, but the Golgi and the mitochondria are the two main ones, along with the chloroplast in plants that we will talk about. The nucleus technically also is an organelle, and the main thing, of course, you need to know about that is that it is the place where you store the chromosomes, the DNA. Alright, here's more of a plant cell, so there is a bit of a difference. This would be considered more of some type of animal. Actually, this has a flagella, so this is some single-celled organism. It's a eukaryote, but single-celled. This would be what a plant cell looks like, and you'll notice these are chloroplasts, and these basically you need to understand how they're different from the mitochondria. The mitochondria, oops, let's change this to a pen. All right, the mitochondria here and the chloroplast here do similar functions, but of course the chloroplast uses light to enhance the efficiency of the recycling of ATP, which I have a whole video on that. You need to watch that one. All right, so this is generally what we want to look at, and next up we're going to be talking about the Golgi apparatus.